Receptors in sensory neurons receive sensory information. This sensory information travels via the peripheral nervous system to the dorsal root ganglia and via the central nervous system to the brain. The axons in peripheral nerves can regenerate if damaged. However, axons in the central system cannot. This leads to permanent disabilities. Sensory neurons provide a useful model to study how to repair axons. After peripheral axon injury signals lead to transcriptional and translational changes to promote axon regeneration. Epigenetic changes also occur. These result in genes involved in regeneration being switched on or off. In addition to the neuronal mechanisms, the surrounding cells assist to reactivate a pro-regenerative program following axon injury. We found that satellite glial cells, which completely surround the sensory neurons, contribute to the promotion of regenerative growth. Our results highlight that the neuron and its surrounding glial coat form a functional unit that orchestrates nerve repair. We're also studying how immune cells, blood vessel cells, and the meninge assist neurons in the axon repair process. We hope to learn how we could apply these mechanisms in therapeutic terms in order to overcome the poor regenerative capacity of central nervous system neurons.